Hello and welcome. In this session, I'm going to talk about a very important genetic event, that is mutations. A lot of information has been generated about this event over the last almost 100 years after its discovery. I'm going to focus today on the very basic aspect of this process, that is what mutations are, defining them very clearly, and how many types of mutations are there, and towards the end, what is the chemistry of mutation. And then in subsequent uh, sessions, I will go about the other aspects of this event. But before going into the details of the main topic, I would like to review or revise with you some basic aspects, which are very important one to remember when we talk about mutations. So if we talk about the background, see, we know that the basis of genetic heredity is the genetic material. That's how Mendel started his concept of Mendel's law of genetic inheritance. But now we know this genetic material is the DNA molecule, which is present in all the cells across the living world, uh, except some RNA viruses. We also know that structure and organization of DNA molecule, which is very well known, is the same in prokaryotes to eukaryotes, from viruses to plants to human beings. There is no difference in organization of this DNA molecule. That is, it has a backbone of sugars, deoxyribose sugar, phosphoric acid, and bases. Now, the variation is in the bases. That is, there are 4 types of bases: purines and pyrimidines, like adenine, guanosine. Pyrimidine is thymidine and cytosine. So these 4 bases, A, T, G, C, basically are the differential factors, you can say, of the genetic material. So it is the arrangement of this ATGC which decides that what a gene is going to code for. Okay. Now, another aspect in living world, each organism has a set of phenotype or phenotypic characteristics on the basis of which we differentiate different organisms from one to another. For prokaryotes, we can't see them, so they are differentiated based on their biochemical nature, what they can synthesize, what they cannot synthesize, what they can use as a food material, what they cannot use. But other than that, the living world can be differentiated based on their features. Okay. Now, these phenotypic features has a basis of or has a genotype. Okay, and this genotype is represented by a set of genes. And each gene has a specific base sequence. That is what I was trying to mention. So phenotype has a genotype, and genotype is governed and decided by a set of genes in all the organisms across the living world. Okay, and the language of the genetic information, now we know, is a genetic code that is in the form of a triplet code. That is a set of 3 bases, codes for a specific amino acid. So all 19 amino acids has a well-defined sequence of codes. So it is very important to remember this aspect, because the main topic which I'm going to talk about ultimately rests here. Okay. Another aspect of this genetic information is the flow of information within the cell. That is, it follows a central dogma pattern. What is central dogma? From DNA to RNA and RNA to protein. So genetic information contained in the DNA first transcribed into RNA, and there are three types of RNA: tRNA, rRNA, and mRNA. And you may be knowing the role of these three RNAs in the second process of flow of information, that is synthesis of protein, that is translation. So you have transcription, formation of these transcripts of three types of transcripts: RNA, mRNA, tRNA, and rRNA. Okay, and then with the help of these three and other, of course, cellular factors, you have the synthesis of protein. Now, a change in this base sequence of this genetic information of any gene, or if a change takes place in the base sequence, it changes the genotype, it changes the gene structure, which has the manifestation on the phenotype, which it controls. So a change in gene sequence changes the genotype, and that change in genotype results into change in phenotype. Okay, and it is this change in base sequence of a gene is called mutation. And the organism or the strain or the member which has this change, we call it as mutant. And the process by which this change occurs from normal to changed, we call mutagenesis. So these three terminologies are very, very important for you to understand. And I will talk about two of them today, that is mutation and mutant. Mutagenesis is the process which helps in bringing about this change. And when we talk about how this change in the base sequence takes place, it is spontaneous, it happens on its own, or it can be induced. So I will touch upon that part later in the subsequent videos. But today, I'm going to focus on very specifically making you understand what this process of mutation is, and how many types of mutations are there, and then what is the chemistry of the mutations. Okay. So if we try to define this process, what are mutations? Okay. So you might have by now got a hint that what I'm trying to refer actually. So basically, one definition which comes into that: sudden, stable, inheritable, transmissible, discontinuous variations which appear in organisms due to changes in genomic sequence. Let me explain what they're trying to put here. That is a sudden inheritable change which is occurring because of a change in genomic sequence. So two things you have to remember: the change is sudden, means it is happening in the current generation. It has not come from the parents. So it is happening in the current individual, and it is passed on to the next generation. So it is transmissible. It is stable and it is transmissible. Okay, and this change is bringing about change in the genomic sequence, is bringing about change in phenotype. Okay, so one definition which one can have for mutation is a sudden, stable, inheritable, or transmissible variation which appear in the organism due to change in genomic sequence. Okay. Or simply you can say that it is the alteration in nucleic acid or nucleotide sequence of a DNA molecule. Now, what is the implication of the second definition? That we are simply trying to say that if there is a change happening in the nucleotide sequence of a DNA molecule, that is mutation. It may have an impact on phenotype, it may not have impact on phenotype. So this is more broad definition for mutations. That is, we include in mutations any change in nucleotide sequence of an organism, which may have a bearing on phenotype, which may not have bearing on phenotype. But change is happening, it is undergoing mutation, and this change is heritable. Keep it in mind that the change is inherited. It is going from one generation to another, from one strain to another strain, it is going. So it satisfies the broader definition or the previous definition that it should be transmissible, should be stable, should be sudden. Okay. So basically, to conclude this part, the mutations are a alteration in nucleotide sequence of a DNA, which are sudden and inheritable. Okay, fine. If we agree to this definition, then the question is, how many types of mutations are there? Okay, so there are many ways by which this event has been classified. Okay, so one such basis is the direction. Now, what I mean by direction? See, you have a population, we call it as a wild type population. Now, in this wild type population, when you observe a variation in an individual, means which is different from the other wild type members, we call that as mutant because a change at the genetic level has happened, and we call it as forward mutation. That is from wild type to a mutant phenotype. For example, in a pro plus population of E. coli cells, if you observe a pro minus phenotype, it means it is a mutant and it is a forward mutation from wild type to mutant type, from pro plus to pro minus. Okay, so it is a forward mutation. And then you have a back mutation or reverse mutation, or the process we call reversion. That is, what is this reversion or back mutation or reverse mutation? Is if you have a population of mutants. For example, if you have a population of pro minus cells, and in these pro minus cells, if you see a pro plus phenotype, it means this pro plus phenotype has come from pro minus phenotype. So, and that is because of a mutation happening into the pro minus population, which has made one of the cells or few cells changing the phenotype from minus to plus. So this is known as back mutation. So now you understand. So you have a forward mutation and a back mutation. And I will today deal with forward mutation. And maybe in subsequent sessions, I will take up the reverse mutation. So to begin with, mutations are classified into two categories, forward mutations and the backward mutations. Forward mutations are the changes taking place in the wild type population with a mutant phenotype. So that is the forward mutation. Fine. Now, what is the other basis of classification? In which type of cell the change is happening? As you know, or you may be recollecting that higher eukaryotes, they have two types of cells: somatic cells, which constitute the body of the organism, and then the sac cells or the germinal cell, okay, which takes part in sexual reproduction. So we have two types of cells in diploid eukaryotes, and in prokaryotes, you have single set of genes or chromosome. There, whatever happens, that happens to that single set of genes, So based on that, you have one type of forward mutation, not like here, because they don't have germinal cells and the somatic cells. There is only one set of cells, and I will talk about that in a while. So, but then one type of classification which is followed is based on the type of cell it is targeting: somatic mutations and germinal mutations, or somatic cell mutations and the germinal cell mutations. Now, the mutations occurring in somatic cells are not heritable. They occur in an individual. They may have a phenotypic manifestation, but then they are gone with the death of that individual. They are not passed on to the next generation. That's one thing which you have to remember to differentiate between somatic types and the germinal cell. But then if a mutation is occurring in germinal cell, that is alone goes from one generation to another. Okay. So that is the difference. The somatic cell mutations, though occur, they manifest also like chimeric patterns in plants. In plants systems, they show invariably this kind of mutations. And also animals, they show this kind of variegation pattern on their skin or in eye colorations. But plants have more examples of somatic mutations. But these mutations, as I mentioned, are not inherited. They are they just end when the individual plant or animal dies. But in case of plants, if a plant can be vegetatively propagated, then it can go from one to another to another. That is another part. But germinal mutations, or the mutations occurring in germinal cells, are alone inheritable. So that you have to keep in mind. Okay. Now, another way of classifying the mutations is based on the conditions they appear. Means there are mutations or the changes at the gene level occurring are showing their effect on a phenotype, irrespective of the conditions a cell or an individual is living or growing. Okay, that is known as non-conditional mutations or mutants. It means non-conditional mutants or mutations are called when a mutant phenotype is seen under all conditions. Okay, here also you have two types of mutations or mutants: null mutations or null mutants, and the leaky mutants. Null mutants is all the individuals having that mutation are shown. That is null mutants or null mutations. And then you have a leaky mutants or leaky mutations. Leaky mutants or leaky mutations are the which have or some members of the mutant population show weak phenotype or slight features of that mutant phenotype of the wild type phenotype. Okay, so some individuals um, show a weak expression of the genes, and therefore they show some characteristics or features of the wild type phenotype. So you have null mutants or null mutations and the leaky mutants or leaky mutations. Depending on how strict the mutation is, how strong the mutation is in controlling the phenotype. So it can be null or leaky. And then opposite to this type of mutation is the conditional mutations or conditional mutants. These terms we can use interchangeably because mutation leads to generation of mutants. Okay, so opposite to this is that certain mutations, when they occur, will show their impact on phenotype under specific conditions. In the sense, if a cell is grown under normal conditions, they do not show mutant phenotype. If the cells are grown under specific growth conditions, then only the mutant phenotype is shown. They are environmental conditions, for example, temperature. So if a mutation is affecting the sensitivity of bacterial cell, for example, to temperatures, then what will happen? Under normal conditions, the cell will grow. But if you increase or decrease the temperature, then it will show that phenotype of sensitivity to temperatures. Or other mutations, for example, suppressors. If there is another mutation which is suppressing the the expression of a actual mutant phenotype, so it means there are two mutations have occurring. Until unless this mutation is present in that genetic background, it is not going to show the mutant phenotype. I will talk about this part towards the end, that what is this suppressor mutation is basically your suppressor sensitive mutations. So for suppressor sensitive mutations, you have to have that genetic background. Then only you will see the mutant phenotype. Under normal conditions, you won't see, though the mutation will be there, but it won't show the mutant phenotype until unless there is a genetic background. We call it as suppressor sensitive mutations. Okay, so another way of classification of mutations is they depend that when they show their impact on a phenotype, means non-conditional or conditional. Okay. Now, another way of classifying mutations is the kind of change at molecular level a gene is undergoing. Okay. So based on that change, we classify them. If we know what kind of change a gene has undergone, either it is point mutation, substitution, insertion, deletions, or the frame shift. I will take one by one all these 5 types of events and explain to you what these they mean actually. So let us first look at what are these point mutations. Okay, so in point mutations, we see that a single nucleotide is replaced. Just one nucleotide is replaced in point mutation. For example, this change can be purine to purine, means A to G, or it can be pyrimidine to pyrimidine. In a sequence, in a gene sequence, if A is there, A is changed to G, or T is changed to C. So the sequence is changing. So where it was A, it becomes G. And if it was T, thymidine, it is changed to cytosine. Single, only one base change. Therefore, it is called as point mutation. One. Or it can be transversion. That is, a purine is changed to pyrimidine or vice versa. Means A or G is converted to T or C. So in point mutation, keep this in mind that there is only change in single nucleotide, number one. Number two, it can be purine to purine or pyrimidine to pyrimidine, or it can be purine to pyrimidine and vice versa. This is the law in point mutations, but only single nucleotide. Keep this. There can be double mutations. Double mutations means where you have two nucleotides changing, in the same fashion. So you can have in point mutation or double mutations, these kind of changes. And then you have insertion or deletion. Now, insertion and deletion also can be of a single nucleotide. A single nucleotide is added or deleted, inserted or removed. Okay. And addition is also changing the sequence pattern. And if you remove or a single nucleotide is deleted, that also results into change of the specific sequence which it was having earlier. Or there can be a large piece of DNA getting inserted or removed from the original sequence. Okay. So there can be an insertion or deletion of single nucleotide, and there can be insertion and deletion of a large piece of DNA. Now that's where the back mutation plays a role, because the insertion and deletion of large piece of DNA or sequences from the gene are basically makes this gene permanently non-functional. There is no way it can go revert back to the wild type. Whereas these addition of or deletion of single nucleotide, they are reversed, or they can be reversed, So and that also you keep in mind when I discuss about the reversions of the back mutations, So anyway, coming back here to this, so point I'm trying to make here is that insertion and deletion of single nucleotide is possible, which brings changes in the original sequence, and that leads to mutation and mutant phenotype, number one. And also the deletion and insertion can be of a more than one nucleotide. Okay, so both are possible and they occur in nature, And then you have the frame shift mutation. Means, see, I told you this addition deletion of single base pair. What happens? That addition or deletion, it affects the genetic information. As I mentioned, this information is residing on the DNA molecule in the genetic code or triplet code. So if you remove or add anything into it, it changes that reading frame, what we call, how the messages are read through, using the mechanisms. So that reading frame is changed. If you remove or add any nucleotide, additional nucleotide, or you remove, then this reading frame is changed. And the consequence of this change in reading frame is that the the these the protein sequence or the amino acid sequence after this mutation, point of mutation, is changed. And the changed protein sequence may make the resulting protein non-functional, means affecting the phenotype. When a protein is not able to function the way it was functioning before mutation, it will show a change in genotype as well as in phenotype. Okay, so this addition and deletion of nucleotide results into change in the frame shift, okay, or the reading frame, basically, okay, and that results into the formation of wrong protein. So, by and large, to conclude this part here, I have tried to put you that mutations based on their direction can be forward and the backward, depending on the condition where a mutation is showing the phenotype or not. Impact on phenotype, it can be conditional and non-conditional. And the kind of change, third way of classifying is the kind of change at the molecular level is a DNA sequence is undergoing. It can be point mutation, can be insertion deletion, and the impact of insertion deletion is the frame shift. Okay. Now, I will close this session now, and I believe that I'm able to reach to you and make you understand this slightly complicated process in a manner which probably you can understand. So before I stop, if you like this video, kindly subscribe my channel and like this video. And I will be coming back to you with other aspects of this process or even genetic event mutation. So thank you very much for patient hearing and your attention. See you soon.